Welcome to this lecture on osmoregulation in fishes. Imagine living your entire life completely submerged in water. For fishes, this creates a constant life or death challenge to keep their internal body fluids perfectly balanced. Today, we're going to explore the fascinating biological machinery and the underlying physics that allow both freshwater and marine fishes to survive and thrive in their respective environments. To begin, let us think about what happens when you sit in a warm bathtub for a very long time. You might notice your fingers starting to get all wrinkly. This happens because your skin is interacting with the water around you, and water is actually moving in and out of your cells through a natural process called osmosis. Now, why does this matter for a fish? Because a fish lives its entire life completely submerged. It is constantly fighting a hidden battle. If it did not actively fight this battle, the fundamental laws of physics would take over. A fish in the ocean would shrivel up like a dried raisin, while a fish in a freshwater lake would swell up and burst like an overfilled water balloon. Now that we understand the intuitive challenge, let us look at the formal biological definition. Osmoregulation is the active, energy-consuming process by which living organisms maintain the constant osmotic pressure of their internal fluids. It is not a passive process where the fish just floats and hopes for the best. The fish must spend significant metabolic energy to strictly control the balance of water and dissolved salts, known as ions, inside its body. It must maintain this precise internal balance regardless of what the external environment is doing, whether that environment is a pure mountain stream or a highly concentrated salt lake. Knowing that this is an active process, we must ask what parts of the fish are doing the work. For fishes, their gills, kidneys, and digestive tracts work together as a highly coordinated biological system. Depending on the environment, this machinery is constantly pumping ions in or out and working to either conserve or excrete water. This active regulation is absolutely critical because it ensures that the cellular environment remains perfectly stable. Without this stability, the vital enzymes and metabolic processes that keep the fish alive would fail to function correctly. Without this machinery, immediate cellular failure and death would follow. Understanding this biological machinery leads us to ask, why does this concept matter so much in the real world? Osmoregulation is the very reason why we have distinct freshwater and marine ecosystems. It explains why you cannot simply drop a freshwater goldfish into the salty ocean and expect it to survive. This process dictates the evolutionary adaptations, daily behavior, and geographical distribution of almost all aquatic life on Earth. Furthermore, this knowledge is incredibly practical. Scientists and farmers in aquaculture rely on a deep understanding of osmoregulation to create the perfect water conditions. This keeps farmed fish healthy, helps them grow faster, and ensures they remain disease-free. Without this knowledge, our global food supply from fisheries and our conservation efforts for endangered species would completely collapse. To truly appreciate the physical forces this machinery is fighting against, we must look at the mathematics of water movement. Water moves across the semi-permeable membranes of the fish's gills due to a powerful physical force called osmotic pressure. We calculate this exact pressure using the Van't Hoff equation. Let us break down the symbols. Pi represents the osmotic pressure, usually measured in atmospheres. The variable I is the Van't Hoff factor, telling us how many individual particles a salt breaks into when dissolved. C stands for the molar concentration difference of solutes between the inside of the fish and the outside water. R is the ideal gas constant, and T is the absolute temperature in Kelvin. Physically, this means the pressure driving water into or out of a fish increases if the water is saltier, meaning a higher C, or warmer, meaning a higher T. Now that we have our formula, let us apply it to a freshwater environment. Imagine a fish swimming in a calm lake at a temperature of 298 Kelvin. We are given that the internal fluid of the fish has a solute concentration of 0.3 moles per liter, while the pure lake water has a concentration of 0. We want to find the osmotic pressure driving water into the fish. First, we find the concentration difference, which is simply 0.3. We use a Van't Hoff factor of 2, assuming the solutes act like a simple salt. Plugging these values, along with the ideal gas constant, into our equation, we multiply them all together. The result is approximately 14.68 atmospheres. Physically, this massive pressure means water is constantly trying to crush its way into the fish's body with immense force. Seeing that massive inward pressure of over 14 atmospheres brings us to an important biological question. How does the freshwater fish survive this? Because the fish is constantly absorbing water through its gills, it is at risk of swelling up and bursting. To combat this, the freshwater fish must constantly pee out large amounts of very dilute urine to get rid of the excess water. However, flushing out all that water also flushes out valuable salts. To maintain its internal balance, the fish uses special energy-consuming cells in its gills to actively pump lost salts back into its blood from the surrounding water. It is a continuous cycle of bailing out water and pulling in salt. Let us contrast that freshwater example by calculating the dehydration threat for a marine fish. Consider a fish living in the ocean at the exact same temperature of 298 Kelvin. The ocean water is very salty, with a concentration of 1.0 moles per liter, while the fish remains at 0.3. We need to find the osmotic pressure sucking water out of the fish. First, we determine the concentration gradient by subtracting the internal concentration from the external one, giving us 0.7 moles per liter. We plug this new difference, along with our constant Van't Hoff factor of 2, into the equation. Multiplying these values reveals an osmotic pressure difference of approximately 34.25 atmospheres. Physically, this means the ocean is acting like a giant sponge, violently sucking water out of the fish's body. With over 34 atmospheres of pressure pulling water out, we must understand how the marine fish prevents severe dehydration. Because the ocean is constantly draining its fluids, the marine fish has no choice but to constantly drink seawater to replace what is lost. However, drinking seawater introduces a new deadly problem. It brings in far too much toxic salt. To handle this, the fish utilizes specialized chloride cells in its gills to actively pump the excess salt back out into the ocean. At the same time, its kidneys work to conserve as much water as possible, producing very tiny amounts of highly concentrated urine. It is the exact opposite strategy of the freshwater fish. Before we visualize these survival mechanisms, let us formalize the terminology used by biologists to describe these two opposite states. Freshwater fishes are described as being hyperosmotic to their environment. The prefix hyper means over or above, indicating that their internal salt concentration is higher than the surrounding fresh water. This is why water flows into them. Conversely, marine fishes are hypoosmotic to the ocean. The prefix hypo means under or below, meaning their internal salt concentration is lower than the highly salty seawater around them. This is why water is constantly drawn out of their bodies. Understanding these two terms is key to mastering the topic of osmoregulation. Now that we have defined the terms and done the math, let us look at this comprehensive diagram to visualize everything we have learned. On the left, we see the freshwater fish. Notice the blue arrows showing water passively entering through the gills due to osmosis, and the large volume of dilute urine being excreted at the bottom. The red arrows show the active uptake of ions to replace lost salts. On the right, the marine fish faces the opposite challenge. The blue arrows show water leaving the body, forcing the fish to actively drink seawater at the mouth. The red arrows here show the active excretion of excess salts through the gills, while the tiny droplet at the bottom represents the small volume of concentrated urine. This perfectly illustrates the two distinct survival strategies. We have covered a lot of ground today, so let us review the key takeaways. First, osmoregulation is the active, energy-consuming process that allows fishes to maintain their internal osmotic pressure against the forces of nature. Second, by using the Van't Hoff equation, we can calculate the immense physical pressures driving water into or out of the fish, proving just how hard their biological machinery must work. Finally, we saw the two distinct strategies. Freshwater fishes are hyperosmotic, constantly excreting water and absorbing salts, while marine fishes are hypoosmotic, constantly drinking seawater and excreting excess salts. You now have a complete understanding of how these incredible animals maintain their incredible balance and stay alive in completely different aquatic worlds.